Yeah, Mac, feel free to start whenever you're ready.
Oh, great. Thanks so much. All right, well, thank you very much for having me today. So my name's Mac. I'm a professor of systems neuroscience at the University of Sydney in Australia. Um, and what I'd like to talk to you about today is some work that my group has been conducting, trying to make sense of an often overlooked structure in the context of the entire brain, known as the thalamus. So it's shown here in blue from these beautiful Golgi stains, a structure that is deeply interconnected with the cerebral cortex, the main focus often of computational neuroscience, but also as an interface between the cerebral cortex and areas like the brainstem, the basal ganglia, and the cerebellum. And so, what I'd like to talk to you about today is how we're going about thinking about this structure and how it's playing a role in both what we call the state and the contents of consciousness. So, what do we mean by different terms in consciousness? Now, it's a bit of a controversial field in large part because people struggle to find definitions of the term that they can actually use in empirical and in modeling studies. And two terms that you might hear when you actually look at the term consciousness in the literature use the verb consciousness in completely different ways. They use it either in what we call the intransitive sense, in which case what we're talking about is consciousness or not. So it's a little bit like the difference between being awake or asleep, or in an anesthetized state versus an awake state. In contrast, we can also use the term consciousness in what we call a more transitive use of the verb. This would be consciousness of a particular thing. So we might imagine that this little dog here is conscious of the food in front of it, the smell of the food, the sight of the food, the fact that it's hungry. So, in colloquial terms, these different terms, intransitive and transitive, really translate quite nicely to the idea of states of consciousness, the difference between being aroused or not, and different contents of consciousness, whether or not we're conscious of a particular stimulus. And what I'd like to convince you today is that this structure of the thalamus is actually deeply related to both the states of consciousness as well as the contents of consciousness. But part of that is actually challenging because if you actually go and look in the literature at what the thalamus is involved in, if you go and survey all different studies in human neuroimaging, both in the context of fMRI as well as EEG, the relationship of the thalamus with cortical dynamics is actually quite diverse. You can find really lovely evidence that the thalamus is actually quite important for boosting activity within particular cortical areas. And this might happen via a particular loop between the cortex to the thalamus that comes back to the cerebral cortex. You can also find really nice evidence that the thalamus is involved in coupling different areas that are actually distributed across the cortex. So you might imagine that there's a parietal area and a frontal area. They have this as a shared projection from the thalamus that can allow them to couple together in particular oscillatory bands, say in the context of an attention task. If you go one step further out, you can actually find that there's really nice evidence that the thalamus, in particular the higher order thalamus, is involved in what we call topological signatures in the brain. So this might be the fact that two different thalamic territories are both supporting different structures in the cerebral cortex, let's say coupling in one domain and then another domain. But importantly, they actually are able, the thalamus is actually to keep those different areas actually separate from one another and allow processing within different domains, different topological domains in the cortex. And you also find as well, quite puzzlingly, that the thalamus is actually involved in promoting variability in its downstream cortical targets. So an area in the thalamus might project to a region in the cortex, and now it actually is able to express a wider dynamic range. So this is a really interesting challenge. How is it that we can kind of take all these different signatures that are related to thalamic activity or thalamic patency and actually understand how they might be involved in these different conscious states and conscious contents? And what I want to argue today for you is that a really nice way to actually make progress on this front is not to kind of pretend that the brain is just a bunch of balls and sticks kind of related to one another, this kind of more kind of abstract perspective. Instead, if we dive in and we actually embrace the fact that the brain is organized across multiple spatial and temporal scales, we can actually make some really interesting progress in understanding how this system might work. So what do I mean by that? I mean that if we go down and look at the cellular level on the left-hand side, we can actually look at different parts of the thalamus, and we can actually look how precisely they're connected in with different specific cell types, genetically identifiable cell types in the cerebral cortex. But we can also zoom out and we can think of the role of the thalamus in the context of the broader brain as a whole. So we can think about these two tensions at the same time. In the one hand, we've got very precise cell types and very specific details about connections between one cell type and another. And on the other hand, what we have is the context of the entire activity of the system and how those constraints work together. And we argued in a recent perspective paper that taking these two perspectives and trying to think about how they relate together and to one another is actually a really great way to start to think about how the states and the contents of consciousness could actually emerge from the distributed activity of our nervous systems. So what we're going to do today is we're going to start down at that microscopic scale, and then we're going to try to come up with some constraints that have been imposed empirically in some rodent studies. We're then going to try to zoom out to the macroscopic scale and then look at some macaque studies, and hopefully together through that process, try to build this intuition about how this system could work as a whole. All right, so down at the macro scale, here on the left, I've got this complicated diagram with lots of different colored cell types. The important thing to note at the start is that there's many different genetically identifiable cell types that all have very particular locations for cell bodies where the cells like to sit in the cerebral cortex, as well as where their dendrites sit, where the projections of those cells that receive their inputs sit in this cortical circuit. And also they have very particular ways of connecting in with different cell types in the thalamus. So why do we care so much about how the thalamus connects with the cerebral cortex? Well, it turns out there's actually really, really great evidence now that a very particular cell type in the cortex, what's known as a layer 5 thick tufted or extratelencephalic pyramidal neuron, is actually deeply related to the states and contents of consciousness. So this cell type is really interesting. It's the biggest cell that sits in your cerebral cortex, and it sits in every single area of your cerebral cortex. So it's in V1, it's in M1, it's in the prefrontal cortex, it's in the insula. Anywhere that we would call a cortical area has this thick tufted pyramidal neuron. In fact, in the motor cortex, it's the main cell type. It's called a Bett cell that projects all the way down through the cerebral pyramids down into the spinal cord and actually coordinates the alpha and gamma motor neurons that control the movement of your upper and lower limbs. So it's really important cell type. It's actually the main, in fact, primary cell output of the cerebral cortex. And interestingly, for our cases, it actually has this really weird anatomical feature, which is that the cell body down at the bottom here, those little bulbs at the bottom of the drawing, actually sit really, really, really long way away from what we call the apical dendrites, where the inputs of that cell come. And it turns out that the massive distance between those two different areas, where the inputs come in at the top and where the actual firing of the cell occurs, where the action potentials occur, is so spread out that it's often the case that the inputs that come to the top of those cells actually don't have an impact on firing. And what we need now is a way to actually couple those two compartments together, yeah, and essentially allow the feedback that comes into the cell to actually change the way that the cell fires. So we can think of this in cartoon form as like a little compartment down at the bottom, which is controlling the output, and then a compartment at the top in light blue that's actually controlling the context in which that output will fire or cause the firing to change. Now, importantly, for the sake of our argument today, the thalamus has actually been shown to play an important role in gating those two compartments. So inputs from the thalamus, in particular a part we call the matrix thalamus, has been shown to come into that intermediate zone and actually help propagate a calcium wave up the cells, up that really elongated dendrite, so that any input that then comes into the apical dendrites can ride this calcium wave down, hit those basal dendrites around the cell body, and cause the cell to spike in what we call a burst firing mode. Now, that's really, really important because it means that the information processing that can occur in that context is far and above better than if the cell had to fire without that particular calcium wave. In other words, any message that that cell's trying to communicate could be communicated far better in the context of that thalamic gated calcium. So Matthew Larkham, a really brilliant physiologist, he's actually an Australian, but he currently works in Berlin at the Charité. Actually showed that if you place an animal, a rodent, under different forms of anesthesia, in fact, you turn off the state of consciousness. What you're actually doing is decoupling these two compartments. You're actually making it so that the inputs to the top of the cell can't influence the outputs at the bottom. So how does this relate to the thalamus? So I mentioned that there's this particular thalamic type that actually projects into those cellular compartments and actually allows those two different domains to couple together. It turns out that the thalamus itself is actually not really a single homogeneous structure. It's not just like one cell type that's just repeated again and again and again that then projects into the cerebral cortex. There's actually a kind of diversity, a spread, or a spectrum of different types of projections that exist within the thalamus. So some really beautiful work that was done back around the turn of the millennium by Ted Jones, an anatomist from New Zealand, showed that if you look down at the in a microscopic level at different cells within the thalamus, you can really characterize these two extremes. In every single thalamic nucleus, there's a sort of diverse spread of these two. Some of the cell types are, some of the thalamic nuclei are dominated by what we call a core type projection, and some of them are dominated by what we call a matrix type projection. So what do those look like? Well, it looks like in the core type projection that you have an input that comes in a very targeted fashion to what we call the granular layers of the cerebral cortex. You think about this as kind of like the input, the main input to the cerebral cortex. And in the case of a thalamic region that you might know about, like the lateral geniculate nucleus, light will come in from your retina, it'll hit retinal cells, that'll then propagate onto what we call the lateral geniculate nucleus, and then core cells in the lateral geniculate nucleus will project on into V1, into what we call the granular layers, and essentially propagate forward the fact that that photon has hit your retina. So we think about this as a kind of way that a message might be passed from the external environment through into the cerebral cortex for further processing. In stark contrast to that particular targeted message passing, the matrix thalamic cells actually have a completely different type of projection. What they do is the cells, axons leave the thalamus and they come up to the cerebral cortex, where this individual cell's axons branch into multiple different targets. And they actually project to both the supragranular layers of the cortex, those kind of areas where lots of those inputs come, where those apical dendrites sit, and areas called the infragranular layers, where those cell bodies sit. And it turns out that these matrix cells actually have a big impact on the way that those layer 5 pyramidal neurons can actually burst fire. So Brandon Munn, who's now a fellow in my group, he was a postdoc at the time, was really interested in trying to take this notion of understanding the specific or core thalamus and the non-specific or matrix thalamus, and asking how their different impacts on this particular layer 5 cell could affect the kinds of dynamics that could come out of the circuit. So what he did was he took a layer 5 pyramidal neuron with all of its complexity, and he distilled it down into that cartoon diagram that we looked at before. So we now have a compartment for the somatic dendrites. These are the cell bodies. A compartment for those apical dendrites where all the inputs are coming in. This is the distal apical dendrites. And then he had this coupling connection between those two, such that we could turn on and off whether or not the inputs to the distal apical dendrites affected the kind of output that we saw in the somatic dendrites. And so specifically what they do in this case is that if inputs come into the distal apical dendrites and are coupled to the somatic dendrites, what you do is you shift from a spiking mode where you have these little individual spikes that could maybe propagate information relatively sparsely to these really dense burst spikes where you now get this really impactful communication throughout the circuit. So Brandon put together a really simple model of this. Essentially, what we use is something called an Izhikevich neuron, which you could think about as a kind of reduction of the Hodgkin-Huxley neurons, in that they don't have all the complexity of all the different types of channels, sodium channels and potassium channels. What they do is they mimic the kinds of firing that can happen in the cells, whether or not it's burst firing or spiking. And what he did was he had the system wired up into this little sheet, and then he controlled the probability of coupling between the apical and the basal compartments. He used this term beta for that. This is the dendrosomatic coupling. And he also controlled the spread of the influence of the dendrites and the coupling. This is to mimic the idea that sometimes the effect of the thalamus can be this really broad distributed impact from the matrix thalamus, and sometimes you can have these more targeted inputs. So this term now, sigma, is now high when we're tapping into the matrix thalamus and low when we're tapping into more of the specific thalamic inputs. And what we wanted to do is we wanted to make contact with a lot of different theories in the consciousness space that have actually made arguments about macro scale signatures of consciousness. So there's lots of different theories out there. In fact, there's more than you can count. You can go and read this really beautiful review paper from Anil Seth and colleagues in Nature Reviews Neuroscience, where they kind of lay out a lot of these different popular theories. But the important thing for the sake of today's argument is that all these theories make lots of really rich macro scale predictions. For example, integrated information theory argues that conscious state and conscious awareness is related to what we call a bifurcation or a broadcast of information across the network that has a particular informational characteristic. It has a high information complexity. The same with global neuronal workspace, but now it's now about this ignition moment that allows the broadcast across the network. And then a favorite of mine back from in the early 2010s from Murray Shanahan argues that these connector hubs that sit in the broader context of the whole network are really crucial for allowing the brain to process information cognitively. Importantly, we have signatures of each of these different measures. So what we can do is we can take Brandon's model, we can sweep these different parameters that we think mimic the effect of the thalamus on this firing parameter in the cortex, and we can ask whether we can recover these different measures. And lo and behold, if we sweep the beta coupling parameter here on the y-axis and the sigma spatial parameter on the sigma axis here on the x, we can then look on the z-axis at the integrated information, the amount of integration in the system, and the amount of metastability. And in each case, we find this really interesting constructive interference where intermediate values of coupling and high levels of the sigma parameter are actually really good at augmenting all three of these different measures. So now these are from completely different theories of consciousness, but the same mimicked neuroanatomical feature of the conscious system is actually able to recreate these different signatures computationally, which I think is a really exciting way to kind of integrate across these different theories and give them a potential neuroanatomical grounding. Brendan went another step further, and I think helped us understand why this might be the case. And I think helped us with a really rich intuition for this idea about why the thalamocortical system, if it's organized the way that it is, where it can help to couple different neuroanatomical compartments in the cerebral cortex, might be informative for different states of consciousness. In this case, what Brandon did was he took two different data sets that manipulated the state of consciousness. The first one did so naturally, and it did so in a series of macaque monkeys who had intracranial EEG placed on one hemisphere of their cerebral cortex, and they were naturally sleeping. And then there'd be a person that would come in and wake them up. They would naturally wake up, and then they'd be in a wake state. So we're going to represent these three different states now as sleeping, waking, while they're in that kind of intermediate stage, and then awake. And then we have a separate data set where people who are undergoing anesthesia had scalp EEG. So now the quality of the data is not as high. The signal to noise ratio is a little bit lower. But what we have now is access to actual phenomenal report in individuals. And so what in this case happened was that people were given an anesthetic, propofol. They were then they were then roused, and they were either reported that they were unconscious during the previous point in time, or they actually remembered dreaming in that previous point in time, or they were just awake when you went to rouse them and say, no, no, I've been awake the whole time. So we have these three different conditions, and here we're going to call these unconscious, dreaming, and awake. Now, what's important for our example here is that Brandon can now calculate those same measures that I showed you before, the integrated information, the amount of integration and participation coefficient, and then a measure of complexity in the Kolmogorov of complexity. And what he can do is he can map those across those different states, and he can put them in this little low dimensional state space. So now what we're doing is we're representing the three different states in each of these cases in this low dimensional state space, right? We've got phi star on the z-axis, participation coefficient on the y, and Kolmogorov complexity on the x. So you can notice already that although there's some approximate overlap here, they're not really exactly overlapping with one another. And we think there's good reasons for that. It could be that something about the higher signal to noise in the macaques or something about the complexity of the human brain means that some of these states aren't exactly the same. But importantly, if Brandon runs a swarm algorithm on these data that actually fits these particular signatures to the state space, the sigma beta state space that he created in our model, now what I'm doing is plotting the best fit of this model in this state space. And what you find is that the same phenomenal states, so unconscious and sleep, are now mapped to this low beta regime at intermediate levels of sigma. You find that the awake state is actually at intermediate levels of beta and high levels of sigma. But importantly, the waking state and the dreaming state have now been completely differentiated. The waking state exists between asleep and awake, right? You might imagine moving through this part of state space to go from being asleep to awake. Whereas if you're dreaming, you might move into a completely different location in state space that has different forms of dynamics that are potentially mediated by different ways of which the thalamocortical system could act. So I think this is actually really exciting and kind of compelling way to take a really rich anatomical story and actually show that you can extend it up to understand the differences between different ways of actually navigating different states of consciousness. Now, we don't just build models so that we can do data fitting. We actually do them because we'd like to understand what's actually happening under the hood. And so what we did here is we took Brandon's model. So what I'm now showing you is a two-dimensional plot of a sleep state and a wake state from the model, right? So all I've done here is just grabbed a sleep state here and an awake state, so different part of that parameter regime. And what I'm doing now is representing anytime there's a spike in a neuron, I'm going to represent that as a little green dot. So you can imagine this is like a little sheet of the neurons in this column here, in this graph, and they're going to spike whenever they get perturbed to go above some threshold. And then orange is going to occur anytime that the input that came into that apical dendrite can cause a burst fire in the cell's cellular compartment. And it really doesn't take a PhD in computational neuroscience to see that there's more orange dots in the wake state than the sleep state. And we kind of expected that to occur because it fits to a higher level of that beta parameter. We expect there to be more bursting because now the apical dendrites are coming and having more of an influence over the cell bodies and therefore causing the burst firing. But what's really interesting is if you play this out now in a movie over time, what you can actually see now is that there's different trains of these different bursts that are influencing which burst happens next in the sequence. And we actually think that this way of coupling the bursts that happen in a particular moment to the bursts that will happen in the next moment is actually a really key phenomenal signature of the way that the cortex is actually changing over conscious states that are associated with awake and arousal rather than sleep or unconsciousness. You'll notice that in the sleep state, you see these trains of green dots, but they don't last as long. In fact, they don't last anywhere near as long as the orange states do. And if you actually go and look at the dynamics of this model, you actually find that the trains of the wake state bursts actually follow a power law and have really beautiful avalanche dynamics that are really indicative of the different type of universal class that these dynamics might follow. So this is actually a really exciting way of thinking about some of the constraints that we should be imposing on the kinds of models that can explain conscious state in humans. Importantly, we're not the first to point at this. In fact, Dan Dennett, who unfortunately passed away a couple of years ago, actually had a really beautiful description of the conscious brain that actually relied on this notion of what he called fame in the brain. But it was really based on this idea that the current conscious state not only was the thing that you were aware of, but it had a huge impact on what you would be conscious of next. And in this way, he actually drew a deep analogy towards the idea of fame. You imagine that if you're a famous person like Brad Pitt or something like Leonardo DiCaprio, the likelihood of you getting the next movie role is much higher because people already know exactly who you are. You've got this really interesting stickiness in time. Another really deep analogy is to the idea of speciation, the idea that individual species in ecology have a huge impact on what the next species will be in the next epoch of time. And that this type of mathematics is actually really important for understanding how the conscious brain actually supports our experience of the world around us. We're exploring that in future work. All right, so far I've been telling you a lot about conscious states. What about conscious contents? So it turns out that in some other work with Nao Takahashi, Matthew Larkham actually did some interesting work on exactly those same cell types. But now rather than thinking about turning on and off consciousness with an anesthetic drug, now what we can do is we can actually, or they could actually do is show that if you stimulated the whisker of one of these rodents while recording from these whole cells in what we call the barrel cortex, this is the main sensory region of cortex that actually relates to the processing of information from the whisker in a mouse. What you could actually do is you could train the mouse to respond every time it noticed the whisker being flicked. And then once it was operantly conditioned to perform this task, what you could then do is you could go through and you could scale the intensity of the stimulus of the whisker so that you were doing a very, very subtle touch to a very obvious touch. And you could go through that entire grade And you could then map out the probability that the mouse would actually respond, and you get this really nice sigmoid curve. But you can actually also show that these cell types, these layer 5 pyramidal neurons, actually show a very similar curve here, a sigmoidal curve. So that if you're not noticing the whisker twig, you don't have any burst firing at all. If you notice it every time, the cells burst fire a lot. And in intermediate regimes, you see this scaling between no firing at all and complete firing. So you have what we call a neurometric curve. Importantly, Matthew and colleagues could then go in and deploy a series of different tools from cellular physiology to actually show that this circuit was causally involved in the perceptual detection of the task. So here's a little circuit diagram. Here's our layer 5 pyramidal neurons, those same pyramidal neurons I mentioned before, and their projection down to the matrix thalamus. And so what Matthew showed was that if you go in and you actually put an optogenetic light that excites the apical dendrites of these cells, you can actually shift what with the detection curve to the left. What that means is now animals needed far less intense stimulation of their whisker to notice that there was actually a response. You could also go the other way. You could put baclofen, a GABA agonist, into those apical dendrites, and you can shift the curve to the right. So with baclofen involved in the apical dendrites, you now need more evidence, more of a whisker twig in order to notice the effect. Importantly for our argument, though, it wasn't just the apical dendrites that they messed with. They actually went and they injected what's called a DREADS ligand into these layer 5 pyramidal neurons. And then they could find all the targets of those layer 5 neurons, and one of which is in the matrix thalamus. They could then put in a particular ligand that was unique to that receptor, right? It doesn't naturally exist in the nervous system, that only turned off that cell type. And importantly, that shifted the curve to the right as well. So in other words, turning off the thalamus in this loop was basically the equivalent of putting GABA into the and turning off the apical dendrites of the system. So you can't think of this layer 5 pyramidal neuron as existing on its own. It exists in the context of this entire circuit, this emergent circuit with the thalamus. So what we wanted to do as a challenge was to take on this idea of taking our model that I showed you before, that we could show could capitulate differences in states of consciousness, and see whether or not it could actually recapitulate these changes in the contents of consciousness. So Christopher White, a really talented grad student in my group who's now a postdoc with the lab, took this little model that we had of the cortical cells and the thalamic cells and added in a little interneuron to kind of make some of the dynamics more realistic. And then he wired them up into this particular regime. So we have lots of cortical cells in a little ring, all of which have a little inhibitory neuron. And then he had a smaller group of thalamic cells that were constraining the dynamics in the cortex. And so Chris's challenge was, can you take these 3 different results from the empirical literature that we can shift consciousness curves to the left and to the right, and can you recapitulate them in your model? And lo and behold, he was able to do this quite successfully. So now if he mimics optogenetic excitation, he can shift the curve to the left. If he puts in a square wave of GABAergic inhibition, he shifts the curves to the right. And if he impacts the matrix thalamus, he shifts the curve to the right as well. Which I think, again, is really exciting because it suggests that we can reduce this beautiful empirical result down to a really core computational circuit under the hood. But Chris wasn't satisfied with that. What he wanted to do was to take that circuit and then ask whether or not it could actually recapitulate tasks that are really commonly used in the human literature to actually investigate the neural correlates of consciousness. So for those of you that aren't familiar, this is a task called the binocular rivalry task. And the idea here is that exactly the same physical stimulus is presented to 2 different eyes. On the left, we have a right grading to the left eye and a green grading to the right eye. But rather than you seeing a blend of brown crosshairs, what you actually see is a red stimulus that then integrates with, then changes to a green, to a red, to a green, to a red, to a green, and switches stochastically back and forward over time. Now, this is a really, really well-known phenomena. It's been recapitulated just many, many different times. In fact, so much so that it has particular psychophysical laws that are associated with it. And so what Chris wanted to do was to take his little model and force it to undergo this rivalry stimulus. So the way he did that was he injected inputs to 2 different sides of the model that had different receptive curves. And he wanted to see whether or not it gave rise to these types of switching dynamics. And lo and behold, without any single changes to the model, the one that fit already to this beautiful threshold detection task, Chris actually found that his model automatically gave rise to this really interesting frustration-related dynamics that you would see in a binocular rivalry experiment. In other words, his little toy guy diagram was able to kind of recapitulate the kinds of psychophysical laws that we see when we undergo these experiments in the lab. In fact, in most undergraduate labs around the world that have psychology students can recapitulate the same effect. Importantly for us, what we could then do is we could go in and make predictions about what would happen if we did those same optogenetic, GABAergic, and DREADS stimuli. And so we can make very particular predictions about what will happen to the psychophysical laws when you actually manipulate the circuit the way that we would like to manipulate it with causal tools. Unfortunately, you can't test this yet because in mice it's actually really difficult to do binocular rivalry experiments for a very simple physical reason. Instead of their eyes being on the front of their head like humans with our overlapping visual field of about 100 or so degrees, mice's eyes are actually placed on the side of their heads, as is typical of prey species, which means that their visual overlap is about 5 degrees or so. That means it's very hard to create that kind of frustration that we see easily in humans. Fortunately, there's ways around this that can use different types of rivalry stimulus, and we're really interested in testing this going forward. But I really like the idea that I wanted to highlight to you that one of the benefits of computational modeling is not just to say, can we recreate what someone's already seen, but can we actually get out in front and make predictions that experimentalists can test in the future? And odds are that this circuit will not be sophisticated enough to capture all the different things that we can do in consciousness studies. But the hope is that we can actually say really interesting things about the kinds of physical substrates that could support consciousness, and then get out in front and understand how our models could be updated and iterated to be better and better explanations of what's going on under the hood. All right, so in the first part, I've really talked to you about this micro scale structures, right? And talking about both the states of consciousness and the contents of consciousness. What about macro scale? What happens if we sort of zoom out to the level of whole brain imaging and then we start to ask questions about how things like local field potentials actually relate to thalamic activity? Well, here we were really fortunate to have a collaboration with another really great group. This here there in the University of Wisconsin, Yuri Salmon's group and Michelle Redenbaugh, who had done some quite beautiful work in macaque monkeys, showing that if you anesthetize these monkeys with an anesthetic agent, they're fully anesthetized. You can then place an electrode in their thalamus in an area called the intralaminar nuclei, the central lateral thalamus, which is a very matrix-heavy region of the thalamus. You can inject a square wave of current and you can actually wake the animal up from anesthesia. They're essentially functionally awake and they undergo a set of reportable behavioral sequelae that are quite easy to spot. You can also show that if you stimulate different areas of the thalamus, like the ventrolateral thalamus or the centromedian thalamus, you actually don't wake the animal up from anesthesia. I will note that there is some controversy here. Other groups have actually stimulated the centromedian thalamus, which is another intralaminar nuclei. It's actually quite parvalbumin rich and sits further back in the thalamus. But they've done so with a much higher current. So it might be that stimulation actually affects the central lateral thalamus as well. But just to give you the confidence intervals around this effect. So what we wanted to do was to try and see if we could recapitulate the main signatures of this effect in a macro scale context. And so Eli Muller, who used to be a postdoc with me, is now running his own group as a fellow at the University of Sydney in collaboration with Stephen Bing Brunton at the University of Washington. Ported some ideas from fluid flow analysis that are really quite beautiful and elegant approaches to trying to quantify what we call laminar and non-laminar flow. So in the context of fluid dynamics, you can imagine if you turn on your tap, the water can come out quite effectively. Or if you imagine watching a river flowing down, there could be nice smooth flow in that river. Whereas if there's a number of rocks and different particles in the river, now all the river flows start to bump into one another and you can get these emergent turbulent flows or eddies and whirlpools and things of that nature. So what we wanted to know was whether or not conscious states are actually well characterized by these two different signatures of flow. So Eli had an fMRI data set from 7 Tesla with a really nice fast temporal resolution. And what he essentially did was just take a really simple approach. He took the data and he learnt what we call a autoregressive model. So this is just what's called an AR1 model. All it's doing is saying, if I'm at a given point in time, I want to guess what will happen in the next point in time for each of my different regions in my system. In our case, we used like a 400 region parcellation from the Schaefer Atlas. But importantly, what Eli does is he doesn't just estimate that over the course of the session. He then goes through and characterizes for each point in time, how well did that prediction actually work? So he has the prediction in gray, and then he has the actual data in the dotted line. And what he wants to ask in each case is, how big of an error was there between what I guessed would happen and what actually happened? And if you plot that over time, this is just shown for an individual subject. Here now I have time in the x-axis running along from left to right. I have the delay in time that I'm going to track forward from 0 to, let's say, 10 time points in the future. And then what I'm plotting on the z-axis here is what's called the mean squared error, or how big of a gap there was between what I predicted would happen, what actually happened. And the important thing to note here is that there's a huge amount of fluctuation between how well you can predict up to 10 time points in the future at different points in the experiment. So if I'm at this particular point here, I can predict extremely well what was going to happen. The brain states were quite stable or laminar. But if I'm at another point here with one of these little yellow peaks, that means that as I started tricking forward through time, my overall guess as to what would happen in the system got really, really bad. I deviated quite substantially from my predictions. And so, just to show you that that's not the kind of thing you see when you use null models, you can randomly shuffle the data and show that your ability to see that error actually jumps up quite high. So it's now, if you shuffle the data, now you've got no ability to predict at all. But importantly, if you conserve the covariance but scramble the phase, you actually find that the data becomes more predictable. So in other words, the real data is sitting between these extremes of completely random fluctuations, but completely stationary fluctuations, somewhere between these really interesting streams of disorder and order. So you can quantify that with a bunch of different approaches. We do some attractor landscape dynamics in our work. I won't belabor the point here, but if you're interested, there's a really rich literature on dynamical systems analysis that characterizes brain state dynamics according to the underlying differential equations that could give rise to the dynamics. And these actually form the basis of a lot of generative models. And so you can think about this as analogous to a little ball rolling around a landscape, right? Where the current state is some point in this landscape. And you can imagine that you could roll into a particular stable location, or it could have a little bit of energy that then pushes it into another part of the state space. So now you can imagine that this pattern that we saw in these horizons that were either really stable or laminar, or actually really unstable and non-laminar, might be rolling into a valley and then rolling out into a flat spot and then back into a valley and a flat spot, back and forward over time. So we can quantify that. Let me just skip through to this next section because I think this is more related to the thalamus. What Eli could then do is we could take the dynamics over time, and he could actually identify points in the time series of thalamic activity that related to activity of the core thalamus, which we think of as really projecting targetedly into the cortex, and that's in blue, and the matrix thalamus in red that has these more diffuse projections. And what he could actually show is that the dynamics of the system actually tracked with engagement of the thalamus, so much so that when you actually looked at the amount of error that we saw over different forecast horizons, you find this massive, massive drop in the amount of energy that it requires to move to a completely different state whenever the matrix is engaged. So this is really a deep intuition that the thalamus itself is not just controlling the microscopic dynamics, but also the macroscopic way in which the brain state as a whole evolves over time. To convince you that this is the case, rather than just showing you some neuroimaging recordings, what we can actually do is build a really completely different type of a model here. So now, rather than building a little microscopic model, right, that mimics the different types of spiking and bursting, now what we can do is we can build what's called a neural mass model. Now, neural mass models take a lot of the detail that we had before of cellular compartments and apical dendrites and all these different complexity, and they put it into this little ball and stick diagrams that still have constraints, right? There's still cortical connectivity that then connects down to the thalamus in the R and SC and SM that comes back to the cortex in loops. But importantly, what we can then do is we can put in these features of heterogeneity, like the matrix thalamus having a broad projection to the rest of the brain, and we can see what those macroscopic dynamics look like. So, for example, what you can do is you can take this model, you can make it anesthetized by increasing the IPSPs in the cortical model using just like a really, really simple formulation for how GABA will affect the brain. And then you could show that the brain essentially sits in this little anesthetized state. And then you can put a little electrical square wave into the matrix thalamus, right? The same thing that Yuri and Michelle did in their experiment. And you can see whether or not you could recapitulate the finding that they have. So in their case, they found that when they gave propofol to the animal, that they had a really, really low alpha coherence in the cerebral cortex. And when they woke up the animal with stimulation, they had this increase in alpha coherence. In Eli's model, we see the same relationship. We see a low coherence when there's no stimulation, and then a really high coherence when we stimulate the thalamus, the matrix thalamus. But importantly, you can go and then calculate all the other measures that you'd see in the literature when people had already had argued that people were either unconscious or conscious. So I talked to you before about things like Kolmogorov complexity and network integration, but there's other measures like susceptibility, the dimensionality of the system, the variance and the synchronization of the system, and its inherent timescale. And in each of these cases, we can recover the same phenomenal relationship where having an anesthetized model recapitulated the unconscious state, and having a stimulated model recapitulated the awake state. So this is actually really nice evidence that you're not sort of stuck with one particular way of looking at the system. Instead, what you're looking at is the kind of broader signatures of how the system could look through different lenses, different types of frameworks. And I think that's actually a really important benefit of these types of models. Like with Brandon's model, we can also understand how this works. So what Eli was then able to do was stimulate a matrix-rich part of the brain and a core-rich part of the brain, and actually show that the kind of traveling waves that occur when you stimulate those two regions are fundamentally different. So if you look at these two maps on the left and the right, what you find is that the little stimulus that occurs in the matrix, it's like dropping a massive rock in a pond. Now a wave spreads across the entire cortex. Whereas in the case of the core, a little rock might propagate to one location, then the next one, then the next one. So if you plot these out now, the time to peak of the stimulus and the different regions of interest that you have in the brain, you can really develop this intuition that the matrix thalamus is allowing parallel circuits to become active at different points in time, which you could then imagine would change the way that the actual dynamics would evolve. Whereas in the case of stimulating the core regions, now you're going to have a very targeted projection, which is then going to cause the brain to change in a really laminar function. So you can show this again empirically with awake and anesthetized systems. So here's that little horizon plot when you're awake. Here's the horizon plot when you've been given propofol. The laminarity is exactly you'd expect, and that's exactly what we see in our model. But crucially, what we want to then do is go and see whether or not this laminarity measure is the same thing we see in the data. So here's what the modeling data looks like. When you've got propofol, your laminarity is quite high. When you're awake, this is your laminarity. It's is a lot lower. There's a lot more error. And then right at that middle zone, when we stimulate, this is the kind of the shift from the propofol state towards the awake state. And this is very similar to what we see in the macaque brain. There's a bigger effect in the matrix thalamus that we think is related to the heterogeneity that's not modeled in the biophysical model. But the important feature is that you can see this recapitulation and reinvigoration of these nonlinear dynamics that are actually occurring in the system. All right, so I know that we covered a lot today, and they were from completely different territories, but I hope that I've been able to give you a flavor of the kind of insights that you can develop when you really embrace the microscale and its complexity of this sort of thalamocortical circuit down at the bottom level, but also think about the macroscopic constraints that imposes over whole brain dynamics. And the important thing that I want you to come away with today is not so much all the details about exactly which cell type the thalamus connects to and with particular laminar or dislaminar dynamics that you see, but rather that you can develop this kind of mesoscale heuristic model of how the thalamus is actually working. We think that there's cortical complexity that allows the information processing to occur. And these different types of thalamic cells, the matrix cells in purple and the core cells in green, can differentially affect the way that the information can process in the brain, such that you can either integrate and distribute information via the matrix, or you can propagate particular pathways of information via the core. And I think this heuristic model of thalamocortical dynamics is actually really important if we want to start to make progress and understand how this system is actually working. Importantly, you can take this steps further and you can actually then say, how does this help us understand different aspects of conscious states? For example, we all know that conscious states often have this kind of gist-like function where we don't quite know what the thing is that we're thinking, but we can feel that we've got the answer on the tip of our tongue. And we think this might be related to the fact that the thalamus has far less cells than the cerebral cortex, such that any information that passes through that thalamic bottleneck will have to be lower dimensionality than the one that could be processed in the cerebral cortex. We also know that our conscious states are extremely selective, and this can be mediated by the fact that every time the thalamus and the cortex communicate with one another, they connect to an area called the thalamic reticular nucleus, which is an inhibitory blanket that essentially inhibits the entire thalamus, or like large parts of the thalamus. And so the idea would be that every time a particular corticothalamic loop became active, they now make it even harder for another thalamocortical loop to become active. And this is going to buy you something that we call divisive normalization in the circuit, I think, quite effectively. And also then that idea of continuity in time, this notion of fame in the brain, that a particular thalamocortical loop that's active, through the fact that it has these recurrent loops, will now make it much more likely that the same thalamocortical loop and ones to which it's connected will be active in the next time point. And I think it relates to this really interesting notion of temporal stickiness. Importantly, there's actually, since we published our perspective paper, there's actually been empirical work that's confirmed that the higher order thalamus, the matrix thalamus, is actually involved in gating conscious perception. This was done in a study, an intracranial EEG study in human participants in China. And they actually showed that through direct recordings of the thalamus and the cortex that on stimuli that were seen rather than unseen, so very similar to that whisker task, but now with a visual stimulus for humans rather than flicking their whisker or their mustache, you find that the thalamus actually becomes active before the cortex and actually is involved in the scene more so than the unseen trials. And we're working on some modeling work now that tries to integrate this particular finding with some of the state-based changes that we've seen before to try to come up with a coherent approach that actually integrates these two together. At the whole, I really want to leave you with this notion that we, instead of just thinking about the thalamus as controlling the states and the intransitive nature of consciousness in the brain and controlling the state of arousal, which has been known for a really long time, I think there's now really compelling evidence that the thalamus is actually deeply related to our conscious perception of contents, the particular elements that we actually perceive in our worlds around us. And I'd like to challenge you to think carefully about this and see how this changes the way that you think about explaining the kinds of phenomena that you're understanding in your day-to-day work. All right, so I might stop there and see if there's any questions from the group. Really happy to be here and happy to answer any questions that you have. Thank you.
Uh, Thanks a lot, Mac. I'm not sure if you heard the applause at the back. So I'll open the floor to questions first from the individuals who are on the Zoom room. If anyone has any question, they can unmute themselves and ask the question. Ooh, you have some interesting stuff about cerebellum also. That's okay.
Oh, yes. Well, we can always, if you want to ask a question, feel free.
Sure. Okay. Okay, no one from the Zoom room. So anyone here would like to ask a question? Okay, you can come up, face, yeah, so that Mac can see you, and then.
Hello, sir.
Hi.
So my question primarily is that within different states of consciousness, one of them that has not been studied as much is flow state, right? Now within that, wherein you have heightened senses of attention and perception, etc., would you consider there to be a difference in transitive and intransitive state, or like would you consider there to be a difference in activation in thalamus within that state specifically as compared to something like meditation, which is a lot more relaxed, etc., etc.?
Yeah, that's a really great question. We've been having some quite interesting discussions and debates in the lab recently about flow states, and we haven't directly contrasted them with meditative states, but I think that would actually be a really fun thing to think about. And there's obviously so many different types of states that are achievable through meditation, whether it's vipassana or meditating on very particular contents and sort of really augmenting your experience of them. And some of those latter ones might feel a little bit more like flow than something like Vipassana, where you're trying to remove your thoughts and sort of watch them passively. So it'd be really fun. I mean, I'm not an expert in that space, but it would be really fun to kind of think through some of the potential predictions you'd make just based on the phenomenal character alone. Towards your broader question as what we think the thalamus might be doing in a flow state, it's a really tricky one. To me, flow is really deeply related to the challenge that you're solving at the time, the task that you're doing. So whether or not you're a rock climber, climbing a rock, or you're in a jazz band and you're playing in a particular band, and you're in the pocket, or you're in a you know playing a video game and completely absorbed and forget track of time. There's something very important about the complexity of the computation that's being achieved, and I think the thalamus is going to clearly play an incredibly important role in that, right? It's going to ensure that you process just the right information, but remove and sort of downweight the non-important information. Let's say I'm in flow and I'm playing a video game, and then my wife calls me for dinner. If I'm fully in flow, I will ignore the call to my wife, much to my chagrin. I'll get in trouble for that, but I'll stay in the flow state. I think the thalamus is actually really well placed to do something like that. But I also think that the cortex and large parts of the rest of the brain, the cerebellum, the colliculus, the thalamus, the basal ganglia, are also going to be really important in arbitrating as to whether or not you value the stimulus of your wife calling for dinner, or you value the goal that you're achieving in the game, or whether or not you have enough skill to actually anticipate what will happen next in the video game versus anticipate what will happen next if your wife finds out that you haven't come for dinner. Arbitrating these things is not going to be, I think, explained by a single part of the brain, but rather the kind of emergent whole brain dynamics. And so we try to kind of keep that in mind as we're sort of trying to develop better and better explanations for this. But we're not at the stage now where we've got very specific predictions about this cell type and this feature of flow. But very interested to thinking about it more.
Great, thanks a lot. So following up on that, so there is some top-down control coming from these, let's say, cognitive control networks, but do we know how are they getting integrated? Again, like layer 5 neurons, but again, specific from what region? So so is it the core region which is getting these projections from these cognitive control, or is it the matrix, and how is it controlling? Have you looked at that?
Yeah, great question. It's a little bit of a tricky area, in large part because these circuits, the way that the thalamus actually integrates both with the cortex, but also with the basal ganglia and the cerebellum, has actually changed quite a lot over phylogeny, such that in rodents compared to macaques and macaques compared to humans, or chimpanzees compared to humans, been this progressive elaboration of kind of frontoparietal cortices, but also medial dorsal and pulvinar thalamus, and also parts of the basal ganglia like the caudate and the cerebellar nuclei and lateral cerebellum. So in that elaboration, the circuits that have changed, we actually don't really have good evidence right now that's at the same quality that we have for, say, mice, as to exactly where each of the cells project to and in which layer. So in mice, you can go in and do like a huge survey in the Allen, and you could say exactly what cell type projects to exactly what layer in this, wild type cell or some, mouse, or this cree line. In humans, it's all diffusion imaging related evidence. And so that's quite, there's a large confidence interval around exactly where they're projecting. And you then don't have the kind of layer specificity that you'd like to be able to make these claims. I think there's some really compelling evidence for particular circuits between cortex basal ganglion thalamus and cortex cerebellum and thalamus that make a lot of plausible sense, but the evidence just isn't there yet. So I don't know if we're at the stage where we could say something about this particular, frontoparietal network or the dorsal attention network or the, default network has this layer of projection or something like that. But what will really help is kind of work like Emily Finn had some beautiful work a few years ago looking at a working memory task with layer specific fMRI. And there's a lot of groups that are doing this kind of beautiful work now that are providing empirical constraints over whether or not different layers of the cerebral cortex are actually important for different features in cognitive space. And so if you could imagine coming up with a bunch of those constraints, you could then say, well, then the prediction would be that it would be a core rich part of the thalamus if it's going to a granular area, or a matrix rich part of the thalamus if it's going to a diffuse supragranular area. And you could start to like use the empirical data to constrain how the brain could support it. I think we're definitely at that stage where we can start asking those questions now and testing them.
Cool, cool. Anyone else? Any questions? Okay, I have a couple more, so I'll just ask myself. So in the stimulation experiment, how was it clear that we are able to stimulate only the core neurons and like only the matrix neuron, not the core neurons?
Yeah, great. Yeah, it's a great question. So this was Yuri Salman's space, and Michelle has a very detailed paper that they put out in Neuron a few years before we did our work with him and Michelle in Cell Reports, and then we actually kind of dive into this in the review paper with Chris and Yuri and Michelle in detail. But what it boils down to is that the type of electrode that they used to stimulate in their experiment and the kind of particular context that it makes with the local areas have a very low current, which means that you're much less likely to have kind of like impact on the broader circuit. Now, you still don't know for sure, and there's still ways that you could imagine that, a phaptic coupling between the stimulation and local white matter tracks, or some kind of like, stimulation that leads to a secondary effect that then affects other nuclei. We don't have detailed biophysical modeling at that kind of level. So it's very difficult to say it beyond the level of if you stimulate an area with a low current in that particular location that we think is where the central atom nucleus stim is sitting, we would see the a phenomena, but we don't see it if you stimulate a different area that doesn't have the matrix thalamus. But it's not an area that's been like, put to bed, right? There's a lot of complexity there, and it's empirically quite a hard set of experiments to do. So I encourage you to read more if you're interested in this space.
Great, great. I think did share that paper where we published it last year on this large scale organization of these higher order networks in the thalamus. So like these default mode network, frontoparietal network, language network, and it all like occurs around the mediodorsal, medial pulvinar region, some part of the
Ah,
anterior thalamus. And I've just
yeah.
like, I think I'll share that paper and share that figure. Maybe we can discuss it sometime later. How they are associated, like how the core matrix divergence and this kind of higher order, lower order network is related. Again, that was based on this precision imaging data, multiple sessions within an individual. We were just using 3T, not 7T. So, but we were precisely able to map along this medial thalamus different higher order networks. And so again, I always had this question of how does it connect to the core and the matrix projections? Maybe
Yeah.
we do not have the resolution, but I think that could be an interesting follow-up to talk more about this, how these networks are
I,
connected and how they are like active.
I mean, I would love that. Yeah, I know I've read your paper, and I mind wandered about it, but I didn't get too much further than thinking it was a really lovely link. To my understanding, I don't know as much about the pulvinar as I do about the medial nucleus. The pulvinar, a lot of the expansions have been in that medial territory, and there's a really nice perspective paper by James Bourne that suggests that the medial pulvinar is actually sharing, in macaque at least, is sharing, and or maybe marmoset, I actually forget exactly which species, is really connected into major hubs of the default network. So, parahippocampal areas and precuneus and things of that, and frontal areas. So there's some really interesting links there. And then the medial dorsal nucleus, some old work from Middleton and Strick shows that granular prefrontal cortex, like area 946, is actually related into parts of the medial and kind of central medial dorsal nucleus. And where they get their projections from, whether that's other cortical areas or subcortical regions like the cerebellum, is a really interesting kind of open question. And I've got different bets that I would make there, but it's kind of hard because the evidence is kind of lagging behind the theory. But I mean, I would love to talk more about that. It's definitely an area of kind of ongoing interest for the lab.
Nice, nice, amazing. So, and then one paper study you were talking about, I was just curious. So, can we use like EEG to track this kind of latent space attractor dynamics and all across different states? So, there is this project going on in our lab where we're collecting this kind of precision EEG data across a whole range of different tasks and want to include even like meditation paradigms and different episodic projection, working memory, cognitive control. So we want to see if we can have this kind of, again, we want to create this data set first, and then hopefully we are able to track. So do you think like EEG data would be enough to kind of track these landscape dynamics?
Yeah, great question. I mean, I'm not an expert on scalp EEG and some of the challenges with processing. And I think an important caveat that we have to keep in mind that's just kind of an inevitable challenge with EEG is that you've got to decide over which portion of the data you want to set as your state space. So do you want to make it in a particular short window where you can maybe collapse the variance and keep it quite stable? Or do you go over long periods of time where now no one particular moment is a good indicator of the entire state? And we've grappled with this a little bit, and Eli in particular did a bunch of work trying to map the attractor landscape dynamics to EEG and found, broadly speaking, similar results. But the problem was that as soon as you kind of extended outside of these sort of shorter windows, you got these massive variance that was very hard to kind of dovetail with some of the stuff that we've seen in fMRI. And because we're fMRI researchers predominantly, we've kind of put that in the too hard basket for now. But I think it could be really cool if you compared it or if you constrained it with really good behavioral data, right? If you had like a really cool movie or some really good cognitive task that you could kind of really feel like you had a good handle on what the states were phenomenally, then you could actually try to make some cool links there. It's not an area of expertise in the group, so we haven't really tackled it.
Cool. Awesome. So, okay. One more question from.
Hello. So
Hi.
my question is that so we were talking about the gating within those cells. So I was interested in like what sort of mechanisms are affecting controlling this gating, what processes and what sort of projections are there to control this. And the second part of the question is that some of the Friston's work was referring to thalamus, or especially pulvinar, as a nucleus which can control the precision of a prediction error. So they introduced that idea of precision and how it controls the gain of prediction error. So are these two things related to each other, you say?
Yeah, no, great question. So, to the first one, the matrix thalamic regions receive inputs from a lot of different areas. They receive cortical inputs. Oftentimes, and not exclusively, the matrix thalamic regions are what are typically characterized as higher order thalamic areas, which means that rather than getting a primary driver from the periphery, like from the retina or the cochlea, they receive their primary driver from a cortical territory, like a layer 5 neuron from a lower level of cortex. And so they're going to be involved in these really complicated higher order dynamics. Like you can imagine the processing that happens in the cortex has gone through a few layers of different processing, and then it hits the thalamus, which then can come up and broadcast. And so, exactly what that's encoding is a very difficult question that is kind of outside the scope of where we're at as a field, I think, at the moment. But it's really fun to think about. There's also major inputs from the basal ganglia in the ventral tier of the thalamus, so in the VM thalamus and parts of the medial dorsal thalamus, that you can imagine the kind of outputs of the basal ganglia that are going to be about kind of valence related arbitration, dopamine and acetylcholine affecting this kind of like competition architecture will then impact whether or not the thalamus could actually go on and downstream affect circuits in this versus that different representation. And then in the posterior thalamus, like in the pulvinar, a lot of the matrix projections are gated by the superior colliculus, particularly the superficial superior colliculus, which has a really interesting kind of integrative role to play, I think, in constraining cortical dynamics. So there's heaps to kind of dig into in that space. And then the third one would be the brainstem. So there's heaps of inputs from different circuits in the brainstem, arousal structures, glutamatergic structures, hypothalamus, things like that, that have a big impact on the thalamus as well. So I think it's really just early days in trying to kind of figure that part out. In terms of your second question about relating pulvinar to precision, I think that some of that work is really beautiful, and I think was really kind of ahead of the game by 10 to 15 years, to be perfectly honest. And we haven't yet tackled the role of the matrix and the core neurons in a context that, like an attentional context that could be related to precision. But Chris in my group has a bit of a background working in active inference and is really interested in trying to kind of connect up these normative computational explanations with these more kind of implementation level explanations, with the hope that the constraints imposed by both ends will lead to really satisfying explanations that then make better predictions than we currently have. But that's a kind of future project. So I don't want to speak out of turn, but I think that work is really elegant and beautiful.
Cool, awesome. Thanks. Anyone else? Okay, Mac, lovely talking to you again. I'll definitely reach out with a lot more questions. We have some data that we're trying to make some modeling out of. We have some MRI meditation data, some EEG meditation data. We're trying to kind of model cohesively again, and all this thalamus work also. So I'll definitely reach out and we can talk more.
I would love that. Thanks for your time.
Thank you. Bye-bye.
Bye.